Hello and welcome back to Immunology, The War is Over, episode 5, where we are going to focus on all things antibodies. Now, antibodies, B cells and plasma cells are intertwined. You cannot tell the story of one without acknowledging the other, which is actually very beautiful. B cells become plasma cells which generate antibodies, and antibodies are potent weapons in our immune system. But because there's so much juicy stuff to cover, we'll be doing this over 2 episodes. In this episode, we're going to focus on antibodies and the B cell receptor, and in the next episode, we'll be discovering exactly what happens when a B cell comes across its matching antigen, including how T cells help in the process. I am so excited to get into this episode, so grab a cuppa and let's jump right in. So, antibodies, also known as immunoglobulins. These are by far one of the coolest weapons in our immune system, but their powers lie in their ability to work collaboratively. They do not work alone. So if you took a pathogen and some antibodies directed against that pathogen and you combined these in a test tube, nothing would happen. The antibody would bind to the pathogen for sure, but on its own, it's not going to destroy that pathogen. However, if you took this same pathogen and placed it into a human being who had antibodies against that pathogen, it's a whole new ballgame, because the antibodies are now surrounded and supported by the rest of the immune system. And this is where the magic happens. So, the antibody binds to the pathogen and encourages the other parts of the immune system, such as complement and immune cells, to come along and destroy that pathogen. So antibodies are like markers which point out targets for destruction. Now, we will be coming back to complement and the innate immune cells in upcoming episodes, but for now, we're going to just touch on what happens when these parts of our immune system come into contact with an antibody stuck to a pathogen. Let's start with complement. Complement is an incredibly noxious part of our immune system, designed to annihilate pathogens. When complement proteins become activated, it sparks off a cascade. At the end of the complement cascade is the membrane attack complex, which punches holes in the microbe, causing it to die. But along the way, complement also generates inflammatory molecules. Some of these function as chemoattractants, which attract immune cells into the area. Others are opsonins, which bind to the surface of the microbe and encourage phagocytosis of that microbe. There are 3 different pathways which spark complement, one of which is known as the classical pathway, and this is the one that is initiated by antibodies, such as IgM and IgD. So antibodies bound to a pathogen can spark complement in order to destroy that pathogen in an absolute jiffy. And at the same time that they spark off complement, antibodies also encourage immune cells to join in the battle, and they can do this in 2 ways. IgG acts as an opsonin, meaning that when it is bound to a microbe, it will encourage phagocytosis of that microbe. And the other way in which antibodies can encourage immune cells to kill microbes is something known as antibody-dependent cellular cytotoxicity, or ADCC. This is when immune cells, such as neutrophils, eosinophils, or natural killer cells, spill their toxic granules all over that organism. And whilst this property of antibodies was designed by Mother Nature to fight infection, it's something that we bank on when developing monoclonal antibodies to treat cancer. If you think about it, just infusing an antibody that binds to cancer cells is not going to shrink that tumour. The reason that these drugs work is because the antibodies bound to the tumour cells activate natural killer cells, which are capable of destroying those cells. And the final thing that antibodies can do is neutralize pathogens. So, say a pathogen requires a particular surface molecule in order to gain entry into cells. If an antibody happens to bind to that surface molecule, it can impair its biological function, rendering the organism less invasive overall. Similarly, antibodies can bind to bacterial toxins, reducing their harmful effects. Now, you can imagine that in order for an antibody to have these effects, it has to be able to bind to the microbe. And so naturally, extracellular organisms like bacteria come to mind. But antibodies also have a role in combating intracellular organisms such as viruses. And we see this in our clinical practice. When we check for immunity against a virus, we'll tend to check the serum antibodies against that virus. And this is because viruses eventually do burst out of cells and into our bloodstream. And in our bloodstream, antibodies are circulating. So when it comes to viruses, T cells are going to be the mainstay for killing virus-infected cells, but viruses in the circulation is very much antibody territory. So, like I say, antibodies are one of the coolest weapons in our immune system. They can neutralize toxins and viruses, activate complement, promote phagocytosis, and promote antibody-dependent cellular cytotoxicity. Antibodies are very potent indeed. So, that's what antibodies do. And their ability to do this is built into their design. And so over the next few minutes, we're going to see exactly what antibodies are made of. Antibodies have 2 heavy chains and 2 light chains joined together like so. Each heavy chain and light chain has a constant region and a variable region. The variable region of both the heavy chain and light chain come together to form a unique binding site, FAB. On the other end of the antibody, we have the constant region, or the FC region, which annoyingly doesn't stand for constant. It stands for crystallizable fragment. Anyway, this is the part of the antibody which interacts with other parts of the immune system. Now, when it comes to heavy chains and light chains, there are a few to choose from, but one thing that they each have in common is their entanglement with the Greek alphabet. There are 5 different types of heavy chain: gamma, alpha, epsilon, mi, and delta, which corresponds to IgG, IgA, IgE, IgM, and IgD, the 5 different classes of antibodies. And there are also 2 different types of light chain, kappa and lambda. Now, you might be thinking, this is a lot of detail. Why on earth would I want to know how these antibodies are structured? But there is always method to my educational madness, and I promise you that if you understand the structure of these antibodies right now, it makes understanding heaps of other things like antibody class switching an absolute doddle. But it also has huge clinical relevance in conditions such as myeloma. So, what I would like you to lock in right now is this. The class of an antibody is determined by the type of heavy chain that it has, not the light chains, not the shape of the antibody, not the size of the antibody, none of that. The class of the antibody is determined by the type of heavy chain that it has. So, lock that in for me, and as we move through this episode, you'll see why that was important. Okay. So in any one antibody, we have 2 heavy chains and 2 light chains, and there will only be 1 type of heavy chain and 1 type of light chain, meaning you'll either have kappa or lambda, never the 2 together. Now, as you can see, there are 5 classes of antibody, which are all designed to work slightly differently. As we journey through this episode, we're going to come back to this table again and again as we learn about the various classes of antibodies and what they do. But I thought we could start with perhaps some less familiar territory. We saw earlier that antibodies have the ability to neutralize toxins and microbes and engage other parts of our immune system to destroy invading pathogens. But what if I told you that antibodies have yet another function as B cell receptors? Get this, IgM and IgD antibodies function as B cell receptors. So, the first place that you will ever see an antibody in your existence as a human being is on the surface of your B cells. Let's unpack that now. B cells originate in the bone marrow, and here they will undergo the early phases of development, maturation, and testing before being released. And as part of this maturation process, the B cell receptor is installed. And this B cell receptor takes the form of an antibody. Now, initially B cells can only generate the Mi heavy chain. So the first ever antibody on the surface of a B cell is an IgM antibody. Now, this is the basic subunit of IgM. There is another version of IgM which is composed of 5 of these subunits, and this big pentamer IgM with the 10 binding sites is the one that we release into our circulation in response to infection. So both of these are still IgM antibodies because they both have the Me heavy chain, but their roles are slightly different. So this little IgM sticks out of the B cell and acts as the B cell receptor. And this B cell receptor is unique. It has a unique binding site which will recognise a specific antigen. And if you watched episode 1 of this series, this is going to sound very familiar. We saw that every T cell has a unique T cell receptor which binds to a specific antigen, and this means that we can recognise hundreds of thousands of different antigens throughout our lifetime. So just like the T cells, the B cell receptor is unique, which maximises the amount of antigens that we can respond to over time. And just like we saw with the T cells, these unique binding sites are created by gene shuffling. So we take a set of genes, shuffle them around, line them up in a row, and then transcribe them to make hundreds of thousands of unique binding sites. And because this process is entirely random, the B cell receptor, just like the T cell receptor, needs to be tested. We need to make sure that this randomly generated B cell receptor is not going to react to self-antigens and cause autoimmune disease. So, before the B cell leaves the bone marrow, the B cell receptor needs to be deemed functional. Sometimes if a B cell receptor is not quite right, then the B cell gets an opportunity to go back and edit that receptor to make it more functional. But ultimately, any B cell which doesn't make the cut will not be permitted to leave the bone marrow. And this process I mentioned of shuffling genes to create thousands of unique B cell receptors, and indeed T cell receptors, is known as VDJ recombination. This is fundamental to creating antibody diversity, and so we're going to unpack that now. Okay, so antibody diversity is created by shuffling genes, and these genes are the VDJC genes, which is why we call it VDJ recombination. So to illustrate this, I'd like you to imagine there are 4 boxes of genes. Each box is labelled V, D, J, and C, variable, diversity, joining, and constant. Now, there are a number of genes in each of these boxes. The constant genes, as you might expect, come as standard. They're all the same. But the VDJ boxes contain a variety of genes. It's more of a colourful lucky dip situation. So, in order to construct our antibody, to make our B cell receptor, we're going to take genes from each of these boxes, line them up, and then make proteins from them. And this is what's happening inside B cells. Immature B cells start off with our basic DNA sequence that is shared by every single cell in our body. But unlike other cells, B cells are going to change the sequence of their DNA in order to create the B cell receptor. We said before that every antibody is composed of heavy chains and light chains, and the genes which code for these are located on different chromosomes. Heavy chains are coded for on chromosome 14, kappa light chains on chromosome 2, and lambda light chains on chromosome 22. And remember that the light chains on any one antibody will be either kappa or lambda, never the two together. So genes are taken from either chromosome 2 or 22. Not from both. Heavy chains use VDJC genes, so genes from every single box, whilst light chains only use genes from the VJ and C boxes. For some reason, they completely miss out the D. So, in our VDJ boxes, there are a variety of genes to choose from in each category. We want to take a gene from each box, line up those genes, and then seamlessly transcribe them into a unique protein. But when we first take the genes out of the box, they come with some extra genetic material attached, and this will need to be trimmed away. And B cells have a specialised set of enzymes to do this, RAG1 and RAG2. RAG stands for recombination activating gene. And you know how much I love when someone names an enzyme after what the enzyme actually does. It's so beautiful. So these RAG enzymes break and join DNA. They take the V, D, J, and C genes, line them up, and chop out any extra genetic material in between them. And the end result of this is the creation of unique DNA segments which code for both heavy chains and light chains. So this process happens separately for heavy chains and light chains, and then the two unique structures are combined to form an antibody which will be placed on the cell surface as the B-cell receptor. Now, of course, sometimes amongst the chaos of making a random heavy chain and a random light chain, you might end up in the situation where these two structures don't come together very well. If that happens, the B-cell can go back to the drawing board and try to fit a different light chain onto this heavy chain until it finds one that sticks. So, say a lambda light chain from chromosome 22 didn't work, the B-cell might try again with a kappa light chain from chromosome 2. But once a functional antibody is generated, the DNA is finalised. There will be no more gene shuffling, no more sussing out the options. The B cell is now committed to making that one antibody with that particular binding site forever. And this process is known as allelic exclusion. Okay, so the first antibody that the B cell will ever make is an IgM antibody. But once this IgM is deemed functional, the B cells will start to express IgD antibodies as well. This is a sign of B cell maturity. B cells which express both IgM and IgD on their surface are permitted to leave the bone marrow and go in search of their matching antigens. Both IgM and IgD on the surface of a B cell have the same unique antigen binding site, and they both function as B cell receptors. The only difference between them is their heavy chains. IgM has the Me heavy chain and IgD has the delta heavy chain. But how can this be? We just saw that those VDGC genes we shuffled in order to create the unique antibody are now locked in place. We can't do any more shuffling. So how on earth do you start making the IgD when the DNA remains the same? And the answer lies in the primary mRNA transcript. Although the DNA sequence within mature B cells is very much locked in, the RNA transcript can be changed or spliced in order to create a different heavy chain. So, from the same DNA code, this B cell will make both IgD and IgM and place both of these on the cell surface. Now, it's not entirely clear why we need both of these on the surface of B cells. Presumably they have different signalling pathways when they come across their matching antigen. But the truth is, we don't really know. Okay, so IgM and IgD are the first antibodies that we ever make. And these function as B cell receptors designed to seek out their matching antigen so the B cell can become activated, proliferate, and turn into plasma cells, which release high affinity antibodies into our circulation. And we'll be covering all of this in episode 6. But before we get there, we still have to fill in this table and get to know all of the other antibody classes. Let's do that now. First up, IgD. So IgD functions as a B cell receptor. It can also be found in our circulation at low levels and likely plays some role in the response to infection. But overall, its main job, by far, is as the B cell receptor. IgM, as we saw, can also function as a B cell receptor, but there is also a bigger pentamer version of IgM as well. This pentamer IgM is the very first antibody we make in response to infection, and that's because B cells already know how to make the IgM. They're basically born with the ability to make IgM, and later, with the help of T cells, they'll be able to make the other antibody classes. But initially, IgM is their absolute go-to. And this big IgM has 10 binding sites, which sounds impressive, but it's actually necessary. These antibodies are binding to 3D structures on the surface of microbes. So an antibody may bind to a pathogen, but it's not going to be glued to that pathogen. And so antibodies are prone to falling off. To get around this, IgM has 10 binding sites so that it has more of a chance of latching onto the target. Later, B cells will be able to generate higher affinity antibodies which bind more tightly to this specific antigen and require less binding sites, for example, IgG. So for any one antigen, we can think of the IgM as being sort of a rough draft, but later the B cells will switch to making IgG, which has a far more refined, deliberate antigen binding site, which will bind much more tightly to that antigen. But until IgG is generated, IgM will be very much on the case, and it does this by activating complement. IgM is less good at directly activating immune cells. You can imagine that because of the way the Fc portions are arranged, that immune cells would find it hard to place themselves in a way that they could interact with these. But that doesn't matter. The ring of Fc regions easily sparks off complement, and then complement brings in the immune cells anyway, so IgM doesn't have to. IgM is the antibody that is first on the scene. But once T cells get involved, B cells will make other antibody classes, the most common of which is IgG. As we mentioned, IgG has 2 binding sites and binds to antigens with high affinity. On the other end, the Fc portion of the antibody can interact with immune cells and complement. And a really cool thing about IgG is that this is the only antibody class that crosses the placenta, which is handy because we don't actually make any of our own IgG until later when we come into contact with pathogens. So having some of mum's IgG good to go offers some immediate protection until we can make our own. And there are 4 subclasses of IgG: IgG 1, 2, 3, and 4, numbered in order of their abundance. And these subclasses have subtle differences in their hinge regions. IgG 1 and 3 have long hinge regions, which make them more flexible and able to bind to complement and immune cells more easily. On the other hand, IgG 2 binds complement less well, and IgG 4 doesn't fix complement at all. And these varied properties of IgG subclasses are used in clinical practice by drug developers. When it comes to making monoclonal antibodies, we tend to use IgG. These have the longest half-life, and so they maximize the length of time in which the drug can take effect. But it's not just the half-life that's important. Drug developers also select the right IgG subclass for the task at hand. If the goal of a monoclonal antibody is to just block a target molecule and impair its function, you don't need to activate complement or immune cells in order to do that. And so you would choose IgG4 to make your monoclonal. But on the other hand, if we're making a drug for use in oncology and you want the drug to bind to a cancer cell and then you want the immune system to see that antibody and destroy the cancer cell, you're going to want to use something like IgG1, which would do a beautiful job of invoking ADCC by natural killer cells. Interestingly, although it's the most potent, we don't tend to use IgG3 to make monoclonal antibodies. Apparently it's quite difficult to make these, but also this antibody is prone to degradation and has the shortest half-life of all of the IgG antibodies. Okay, so that's IgG, the most abundant antibody in our circulation. The next antibody to touch on is IgA. This is the second most common antibody in our circulation, but the most common antibody found in human secretions. We're talking saliva, tears, mucus, breast milk, you name it. You'll find some IgA antibodies in there. And so IgA is the first point of defense in areas exposed to the outside world, such as our respiratory and gastrointestinal tracts. IgA has 2 subclasses, IgA1 and IgA2. IgA1 has a flexible hinge region and is vulnerable to cleavage by pathogens. You'll see this IgA mainly in our bloodstream as a monomer. IgA2, on the other hand, is a more durable, rigid structure. This is the one we see in our secretions, and it tends to exist as a dimer with 2 antibodies joined together. And central to this joining of the 2 antibodies is the secretory component. This sounds fancy, but it's basically just a molecule made of sugar and protein, which joins the 2 IgA molecules together, but also provides added strength to the molecule, preventing its breakdown, and helps the IgA to anchor to epithelial linings. And IgA is pretty handy. It's able to neutralize toxins and trap organisms. So IgA might even stop an infection before it gets started. When IgA binds to a microbe, it may do this using its dedicated binding sites, just like the other antibodies do. But IgA also has another trick up its sleeve. These antibodies are generously coated with glycans, which can adhere to and trap pathogens. IgA antibodies bound to a pathogen can activate antibody-dependent cytotoxicity by neutrophils, but they don't tend to activate complement. So that's IgA. And lastly, we arrive at IgE antibodies. These are the least common of the antibody classes, but they are very potent and definitely worth a mention. IgE tends to join forces with mast cells. So when IgE is released into the circulation, it will travel around and find a mast cell to hang out with. It will embed itself into the mast cell, and the mast cell loves this because it now has an antenna that it can use to look for antigens. So if an antigen comes along and binds to one of these IgEs on the mast cell will register this signal and degranulate its toxic contents all over that antigen. Now, this system was designed by Mother Nature to help us fight parasites. But of course, the most common place we encounter IgE in mast cells is within allergic conditions such as asthma. So that's the 5 antibody classes in a nutshell. And whilst I've discussed each of these antibodies in a singular fashion, the truth is that antibodies are much more potent when they come together in immune complexes. If you think about it, a single antibody bound to a pathogen could easily be missed by complement or immune cells floating by, but a big complex of antibodies bound to an antigen is going to attract much more attention. So that's a wrap for this episode. Thank you so much for joining me. I hope you enjoyed learning about antibodies and the B-cell receptor. I hope you'll join me for the next episode when we'll be exploring what happens when a B-cell comes across its matching antigen. I'll see you there. Bye.